Well, greetings and salutations. I'm Dan Kastner. I am the Scientific Director Emeritus of the Intramural Research Program of the NHGRI, and it is my enormous pleasure to welcome you today to this, the final installment of the NHGRI mini-series, Bold Predictions for Human Genomics by 2030. And this is a series that actually began, of the NHGRI mini-series, Bold Predictions for Human Genomics by 2030. And this is a series that actually began, if you can believe it, on February 1st of 2020. February 1st is my wife's birthday, actually. I should just acknowledge that fact. And in any case, we have now had 9 presentations on roughly a monthly basis, beginning last February with an interruption last summer for a little bit of time off and an interruption over the holiday season. But we are now at number 10, the splendiferous number 10 in the list. And so in any case, this is just part of what the NHGRI is doing to talk about and observe the strategic vision that was rolled out basically in late October of 2020. And this was our strategic vision, the NHGRI strategic vision. It's the third one that we've had since the completion of the Human Genome Project. And each of these is really a major undertaking in which the Institute reaches out to our constituencies and has workshops and listening sessions and various meetings to talk about the future of genomics, bearing in mind that NHGRI cannot do it all, and that NHGRI is just taking a role to try to lead things and be the so-called forefront of genomics. And so there was this paper that came out in Nature in late October of 2020 that had 4 major components to it, one of which dealt with the principles and values associated with the Genome Project, a second part of it that dealt with building a robust foundation for genomics, the third of which dealt with breaking down barriers amongst the silos that we have, perhaps, or potential silos that we have in genomics, and then articulating some major projects for genomics going forward for the next decade. But the fifth component, which is relevant for this series, is the component that had to do with the bold predictions for the next decade. And the whole idea here is simply to engage people, to get us thinking, to have us imagining things, because the imagination is so important. And, you know, when you think about it, we can kind of predict maybe what's going to happen for the next 5 years, but for the next 10 years, you know, you have to really let your imagination run wild. And if, for example, if one were to have said close to the end of the completion of the Genome Project that within a decade it would be possible to sequence someone's exome for only $1,000, which basically happened within a decade of the completion of the Genome Project, people would say, no way. But, you know, it happened, and it's just by imagining these things that people can set those kinds of goals. And so we really think that this is an important exercise. And we have these 10 sessions. This one is the 10th. And what we try to do for each of these sessions is to pair a couple of speakers, not people that are going to be bickering with each other. We don't want that. But instead, we want to have people that have complementary views and that will basically allow us to think in various different ways and where we can have a civilized conversation. And each of them will give approximately a 25-minute talk under threat of who knows what if they go over. And then there will be a moderated discussion at which I will grill them with 20 questions that I came up with yesterday. We may not get to all 20 of those questions, but we'll try to play 20 questions anyway. And then my colleague, Gunter, we'll have a Q&A with the audience. And so you all should know that if a question pops into your head, you should use the Q&A function of Zoom in order to enter your question, to register your question. Now, if you get called away for something, if somehow, and I don't think this is going to happen, you fall asleep during this presentation and you want to pick up where you left off, all of these sessions are recorded and will be available by Zoom. All of the previous 9 are available. And in fact, you can go back to the very first one from February 1st of last year and hear Eric Green introduce the whole series, and then to hear the subsequent ones as well. Now, today, we are going to be going into the prediction number 10. And that is, just for those of you who need larger print, breakthrough discoveries will lead to curative therapies involving genomic modifications for dozens of genetic diseases. That is the bold prediction. And, some would say, actually, and I have heard already, some saying that this is not bold enough, that we in NHGRI are kind of wimpy. And I think that we may hear this from our speakers. And if they want to argue that point, good. That will make for a fantastic discussion. Our 2 speakers are going to be, and here, we're just trying to mix things up a little bit so that you'll pay attention. Although he's listed second, in fact, our first speaker is going to be Dr. John Tisdale from the National Heart, Lung, and Blood Institute Intramural Research Program at the NIH. John is a senior investigator with the NHLBI and is really a world's expert on bone marrow transplantation, gene therapy, gene editing in sickle cell anemia. One of the real classic diseases, but one that still has been very resistant to coming up with targeted therapies. And so it's really appropriate that we hear about this from John. And then our second speaker is going to be the irreplaceable Tim Yu, who is an associate professor of pediatrics and neurology at Boston Children's Hospital and Harvard Medical School. He is an expert on autism spectrum disorders and neurodevelopmental disorders and on genomic targeting for therapeutics in rare diseases. And so we have really a spectacular combination of speakers. And I think at this point, I have overstayed my welcome, and it is time to turn things over to the good Dr. Tisdale. John, take it away.
Well, thank you, Dan, for this enthusiastic introduction to this really terrific series. I am going to go ahead and start off by saying that you guys were not bold enough. Because I can think of not just genomic modifications for dozens of genetic diseases that are currently being studied, but probably now approaching a dozen ways to look at curing sickle cell disease. I'll show you a few of those as I go. So the rationale for developing genomic therapies for sickle cell disease is, as you mentioned, it's a disease we've known about for more than a decade, the molecular basis known for more than a century, half a century. Linus Pauling describing this as a, quotes, molecular disease. It results from a single substitution of position 6 of the beta globin chain. It's pretty much the same mutation in everybody. There's some compound heterozygotes. So it makes it easier to think about because it's, just one gene we have to go after. This results in abnormal red cells that have a hemoglobin inside that's prone to polymerization on deoxygenation. This causes very fragile red cells, severe anemia, frequent severe pain because the hemoglobin polymerizes, preventing these red cells from going through the circulation, and repeated events like this causing organ damage. It's a very rare disease in the U.S., but in the world, it's actually quite common. In sub-Saharan Africa and India, there are many millions estimated. And current therapies are really limited. It affects virtually every organ system, and in fact, results in a mean age at death of 39 in a recent series published in 2006. Published in 2010, looking up to 2006. But one thing that's worth noting is in yellow here in the 70s, the mortality for children with sickle cell disease was around 25% by the age of 18, but just with screening. So genetic screening in all kids now, you identify them, you put kids with sickle cell disease on penicillin prophylaxis because of a risk of infections with encapsulated organisms because of splenic atrophy. You get virtually all kids to adulthood. So that's one of the first genetic steps taken in sickle cell disease that's had a big impact. But I want to push you a little bit now and say there's a cure for sickle cell disease. It's been around for a long time, and it's called, I'm going to call it that today, whole genome therapy, okay? So this patient I met at St. Jude, she got whole genome therapy in 1984 when she was at St. Jude, had a refractory acute myelogenous leukemia, got a bone marrow transplant from her sibling. It cured her leukemia, but she also got the entire genome of her sibling among her hematopoietic stem cells. They made normal red cells, so it also cured her of sickle cell disease. So this is the kind of work that I've been doing, for now 25 or so years at the NIH, using bone marrow transplants as a way to cure sickle cell disease. So we can either think about replacing bone marrow, as was the case in this individual that I just showed you, or repairing bone marrow of the patient so that we could get normal red cell production, because the stem cells, the seeds of the bone marrow, provide all of the blood for the life of the patient. So these are the current treatments for sickle cell disease. We have one drug that's mostly used, hydroxyurea, and all of the drugs that are being tested now have as their mission to prolong the time between deoxygenation and sickling so that the red cells, instead of getting clogged up as they are here, get through the circulation and back to the lungs before polymerization and blocking of the circulation. We also have allogeneic transplant, which I just told you about. But we can think about taking the patient's own cells and adding a gene with the virus. It's gene addition gene therapy, or using CRISPR, for example, an edited gene that represses fetal hemoglobin and switch back to fetal hemoglobin production from beta S. That would also be, helpful. Or we could think about, correcting the genetic mutation in those cells and giving them back. And finally, it'd be really great if we could do this all in vivo without having to take the organ out, manipulate it, and give it back as a transplant. So we initially showed now some time ago that we can cure patients with sickle cell disease even without replacing all of their marrow with a chemotherapy-free transplant regimen. It worked in 9 of 10. Then we extended it to 30 patients because the patients remained on immunosuppression with this protocol as it was originally written. And in those 30 patients, we were able to wean the vast majority of these patients off of immunosuppression, and they maintained a mixture of their donor cells and their own cells that was sufficient to correct the disease. So all the red cells would come from donor. And that set the stage for us to think about doing half-matched transplants, because most patients don't have a matched sibling in the family. And about a decade later, we looked back to see how these transplants had gone. And in blue, you can see the matched sibling transplants in about 1,000 patients that we found that were reported to the Center for International Blood and Marrow Transplant Research. So all transplants get reported there. So we can have a look at how we're doing in general. You see event-free overall and event-free survival highest for a matched sibling, but even with half-matched or matched unrelated donors, the results are looking really quite good from a transplanter's perspective. And of course, the results are best in kids. That allowed us to ask the question, okay, how much do we have to fix this disease? We transplanted over 100 patients. We had many patients, over 60, with extensive long-term follow-up, where we looked at how much of the donor was in their white cells, the neutrophils, which have about a 2-3 day half-life, versus their red cells. And we identified 3 patients who, despite having a really robust engraftment initially, had a decline over time. They had sickle cell trait donors, so the carrier. And when these patients had 20%, when they crossed the threshold of 20% of their neutrophils coming from donor, we started seeing an increase in their hemoglobin S and a return of their symptoms. And we wondered if this was just because the lifespan of the new red cell is so much longer, it outcompetes the sickle cell. And so Stefan Cordes and Courtney Fitzhugh developed a mathematical model to predict what we would see if it was only lifespan that was driving this need for only 20%. And here you can see the chimerism falling in these patients to the 20% mark here, and then the hemoglobin S going above 50% at that point. And this is the predicted line here showing that it's really just red cell half-life. So that asks, then we ask, can we do this with gene addition as opposed to whole genome therapy, as I was calling bone marrow transplant previously? So in this, we would just add a gene, a correctly spelled beta globin gene in the case of sickle cell disease, put it into stem cells, and then give that back as a transplant, the patient's own cells. With the pretty stringent requirements for correcting the hemoglobinopathies, it has to be high level, sustained, restricted to the red cell, enough to overcome the hemoglobin S that we haven't altered. It has to be self-inactivating this vector system so we don't turn on genes next to where the vector lands. And furthermore, if it's tissue specific, that'd be even better because it's only being expressed in a red cell, which then ejects its nucleus. And that was finally achieved through the use of HIV as a vector system. This is Michel Sadelain's first demonstration in beta thalassemia, which is a related red cell disorder where they're not making a pathologic hemoglobin, they're just not making beta globin, and therefore red cells don't get produced and they have to be transfused for life. And this is Philippe Labouche's demonstration in 2 different transgenic models of sickle cell disease. So we went to our non-human primate to test this. And then we, this model that's been so beneficial for us testing gene transfer approaches was restricted in its use for HIV vectors because HIV does not infect old world monkeys. And we spent some time figuring out what the main restriction was, and it's TRIM5 alpha, which targets the capsid for degradation. So that made it easy for us. We just switched our vector out to have a simian capsid, which won't get recognized, and it'll behave like an HIV vector in human cells. And in fact, we got very high levels of transfer in animals where we competed chimeric vector transduced cells here shown at 30% versus standard HIV vector cells, which were around 3 to 5%. And that allowed us to comprehensively test beta globin vectors. And now we have a very promising forward-oriented beta globin vector with all the bells and whistles to drive erythroid specific expression, where we can reliably now get high-level engraftment and expression of a beta globin construct in the non-human primate. So that set the stage for starting a trial looking at HIV-1-based gene therapy in sickle cell disease. We did this in partnership with Bluebird Bio. And in this study, patients have their cells collected, transduced with a vector expressing a T87Q variant of beta globin, which we can easily see by HPLC. They're frozen, and the patient comes back to get their bone marrow ablated with chemotherapy, the cells infused, and then hopefully they reconstitute with normal red cells. This study went through several groups as we refined both the patient treatment, the manufacturing of the cells, and finally the source of the cells to mobilize peripheral blood, where we got much higher cell doses. We've just published these results now at the end of last year, and I'll show you some of the unpublished data that we couldn't squeeze into here. First is that vector copy number. So this is the average number of vector present per cell improved quite substantially in group C to over 1. So, this on average, each cell has a copy of the vector in group C. And the other thing is that this is very stable over time. What you get in the beginning is what you keep over time. It's much higher than what we saw in group A patients at about 0.1. So this would be the equivalent of, 10% of cells having one copy. But again, very stable over time. So what we got at the beginning is what they kept. This shows the hemoglobin fraction in red coming from vector. So it was modest in group A, 10 to 15% of the total hemoglobin coming from the vector, whereas in group C, it's about half. So this looks like sickle cell trait, the carrier state. And furthermore, the anemia improved. So these patients now have a normal hemoglobin. This shows their pain events before. So they were having, on average, 3.5 pain events that required hospitalization for intravenous narcotics in the 2 years before consent, and none in the subsequent follow-up after transplantation. This is very similar to what we see in the allogeneic transplant setting. So these are our own patients that Alexis Leonard analyzed. And we actually see more pain in the first year in our patients, but virtually no pain in the second year, shown in red. 3 patients only with pain events in the second year. We did have a safety signal in group A. Unfortunately, in 2018, we had one of our patients develop MDS and it progressed to AML. He ultimately died of this leukemia after a successful haplotransplant. But there was no vector in the blast, so we attributed this to busulfan conditioning. But last year, we had a patient develop acute leukemia, and there was vector present in the leukemia cells. The integration was in VAMP4, which is no reported role in cellular proliferation or oncogenesis, and the insertion had no impact on gene expression. And both patients, in fact, had classic driver mutations. They both had monosomy 7 and mutations in RUNX1 and PTPN11. And we recently reported this exhaustive evaluation of this leukemia, attributing it also to disease and transplant, but not to vector. We've in fact seen MDS AML documented across allogeneic transplant regimens. So with whole genome therapy, as I called it, we have a similar frequency of MDS AML across the board, but this seemed to have improved when we improved engraftment rates. If you see in the haplotransplant, we're only about 50% engrafted initially. We had a very high rate, and we're not seeing that now, and the regimen has improved. And Courtney Fitzhugh has been looking more into this, but it looks like the MDS AML is more common in patients with graft failure. So we started to look at our own results in the gene therapy space to see is this transplant related proliferative stress, continued hematopoietic stress due to minimal clinical benefit, underlying risk of malignancy, which we already know sickle cell subjects have. And we were hopeful that in group C, with the higher dose, the higher vector copy number, and the higher expression with complete resolution of disease, that we'd see more contribution by integration sites over time with a reduced proliferative hematopoietic stress. So we looked into integration sites, and this is typically how it's done. We look at where the vector lands, and we express the top 10 here in the bottom, and that's where most of the attention goes. And this provides a unique fingerprint of the original transduced CD34 cells, and we can trace back to see if any clonal expansion is related to any specific integration. So here's integration site analysis in group A, top 10 in 3 patients here versus group C. You can see already that's a much lower fraction representing the top 10, and in fact, a much higher number of unique integration sites, a log higher in group C. This unique integration site number correlated with peripheral blood vector copy number and also T87Q percent over time, so higher integration site number, better. Also higher integration site number, more pancellularity, so up to 90% of the cells containing hemoglobin from vector, and inversely correlated to an ex vivo sickling assay. So we think that this higher dose and higher expression and better resolution may have reduced proliferative and hematopoietic stress, and that may be one way to mitigate the stress. Now, Courtney went back and looked at 2 of her patients for whom she had the mutation identified and found P53 mutations in those 2 patients at diagnosis, and she and Chris Horgan developed a sensitive PCR analysis to go back and look for those mutations to see if they predated transplant. And in fact, both of these patients had P53 mutations at a variant allele frequency much lower than detected at the time of AML, but suggesting that there's also underlying risk based on pre-existing clonal hematopoiesis. And this has been shown by Ben Ebert in lymphoma patients who get autologous transplants, where there's a 14% 10-year cumulative incidence of treatment-related myeloid neoplasms in patients who've been identified as having a clonal hematopoiesis before transplant versus only 4.3 in those who don't. This has also been recently reported in ovarian cancer. So to mitigate this risk of malignancy, in this one approach, we can do screening to see if there's increased risk of hematologic malignancy already present or pre-existing genetic alterations already present in our patients, and do the best that we can to silence the disease after the treatment. And we're hopeful that that's indeed what we've done. We've just reopened this trial now. And in fact, I didn't mention, but in an ongoing trial for thalassemia, using the same vector system, was recently reported. The majority of these patients are now transfusion independent, and there have been no myeloid neoplasms reported in this cohort of patients. Well, we also have, with the advent of the genome now, ways to do genome-wide association studies to identify potential targets for gene editing, for example. And my colleague Swee Thiem, when she was at King's College London, identified a quantitative trait locus that influences F cell production. So BCL11A, when mutated, is associated with higher levels of fetal hemoglobin. So we began with the obvious question: can we inhibit BCL11A or cut its erythroid enhancer to reactivate fetal hemoglobin and switch from beta S that causes sickle cell disease back to hemoglobin F as a treatment for this disease? I mean, we already know that there are some people walking around with mutations that make their fetal hemoglobin persist, and with sickle cell disease that have virtually no symptoms. So this has recently been shown by David Williams' group that you can post-transcriptionally silence BCL11A with a smear and a lentiviral vector and make fetal hemoglobin go up and really have a large impact on this disease. These are some of the data from that publication. You can see their hemoglobin's improving to the normal range. And importantly, the percentage of fetal hemoglobin in the 20 to 40% range long-term. What about cutting instead of using this smear? We have, CRISPR now. Maybe we can cut BCL11A erythroid enhancer, or even cut and repair. So Salome Demirci and my group started off looking at this in the non-human primate model, where we can model this, and he developed CRISPR reagents to cut the BCL11A erythroid enhancer. And at first, he competed in an engraftment study in the non-human primate cells that had the AAVS1 site cut, so we would think that this would not influence hematopoiesis, and that way look to see if there was any deleterious effect of eliminating the erythroid enhancer. And in fact, these animals had normal appearing erythropoiesis. The enhancer edited cells and grafted at the same rate as you saw on the last slide. And in this slide, I show an animal that only got BCL11A enhancer edited cells, and 80% of the cells have edits, and the reticulocyte hemoglobin is comparable to transplanted and non-transplanted controls. We got better as we went, so you can see higher levels of fetal hemoglobin in this last animal that I showed you, and in fact, the indel percentage correlated with the fetal hemoglobin induction that we saw. We've gone on to cut in two areas of the BCL11A erythroid enhancer with Dan Bauer, the plus 55 and plus 58. And now we have quite robust fetal hemoglobin. These are control animals, and these two animals with both sites edited from 30 to 60% F cells and from 15 to 40% fetal hemoglobin, which rises from to quite high levels with phlebotomy. So we mimic the disease state with phlebotomy, and you see the fetal hemoglobin levels going up quite high. And then if we add hydroxyurea, the one drug that we know increases fetal hemoglobin, they go even further. So these are quite high levels of fetal hemoglobin. And in fact, now this has been shown with a CRISPR therapeutics trial with Vertex Pharmaceuticals to result in a similar outcome in patients. So this is a patient with thalassemia. All these dots are transfusions before the treatment. And here you can see the hemoglobin going to normal, and virtually all hemoglobin F in this individual at 14.1. And then in a patient with sickle cell disease, these are pain events and these are transfusion events here at the bottom. And you can see about half of the hemoglobin coming from fetal and a normalization of the hemoglobin. So this too are working. What about fixing by HDR? I mean, that's much harder. It's always easier to break things than it is to fix things. So, you know, in all the stuff that I showed you before, we're just breaking some gene here. Fixing is going to be more difficult, I think. And we've been working on this. And if we take sickle cell disease cells and we electroporate them with, you know, CRISPR, cutting at the beta globin misspelling, and adding template DNA, you can see we can grow erythroid cells, pellet them, do hemoglobin electrophoresis, and we've switched from here the no electroporation control showing hemoglobin S to virtually all hemoglobin A, even if we only have about 30 to 40% correction at the DNA level, because most of the rest of it is indels, and those don't really contribute because we've made them more or less thalassemic. And just for confirmation, this is reverse phase HPLC. You see the beta S peak going nearly completely away here and being replaced by a normal beta globin peak in these two samples. So we try to model this in the monkey. And of course, we don't have monkeys with sickle cell disease. So what we do is just a reverse correction. We introduce the beta S mutation. This is work with Naoya Uchida and Bob Donahue, with whom I've been working for many years. And here you can see in the product shown in blue here, the creation of the sickle mutation, somewhere between 30 and 60% if we use 53BP1 as an adjuvant. And in the second animal, we had two products that we tested, and again, 30 to 60%. And here you can see it by HPLC, by hemoglobin electrophoresis. In the rhesus, we always get some hemoglobin F, but this is A and F without electroporation, and with electroporation, F and S. So we've switched completely from A to S in these animals. What we saw, unfortunately, is that even though the indels persist, and they did for BCL11A, the ones that have undergone HDR drop quite substantially. So we're only at a few percent now. We're trying to figure out how to get better engraftment by cells that have undergone HDR, or maybe get more into the hematopoietic stem cell compartment. But we do see some readout of hemoglobin S in these animals. It declines over time. So this is work that we still need to do. So in the last few minutes, I have just a few minutes until I'm at 25. I just want to respond to a comment from my colleague Suile that I mentioned before. You know, she said we need a imatinib for sickle cell disease. And for those of you who don't know what imatinib is, it's a drug for a leukemia that, you know, has a translocation BCR-ABL that turns on a kinase, and this kinase inhibitor can just shut it down. And if we want to reach all these people, it'd be a lot easier to have a pill to do genome therapy than to have, you know, these more complicated things that I've been showing you. And when I came to the NIH, I mean, CML was the prototype for allogeneic transplant. There was an immune response from donor that would just eradicate the leukemia. It was really remarkable. And this just shows you some data over the years. You know, we had busulfan, hydroxyurea treatments many years ago, started doing interferon, and then started doing stem cell transplants. And this is a really nice bar for a stem cell transplant, for an allogeneic transplant, 60 to 70% long-term survival. But when a kinase was identified and tested in an oral formulation, you can see here now that this imatinib produces a really nice long-term survival in CML. And this has been duplicated over and over. So we really need something like this, a pill you can take. So Björg Goodman's daughter had been working on POG-Z. She discovered that this is required for silencing mouse embryonic beta-like hemoglobin and human fetal hemoglobin. And if you knock out POG-Z, you can get nice erythroid differentiation, knock down POG-Z, and fetal hemoglobin goes up. So this this looked like something that we could think about doing in humans. But transcription factors are difficult to target due to the nuclear localization function. So I said, go find a kinase that's downregulated on loss of POGZ. And so she did this in fetal liver knockout cells, and also look for kinases that were differentially expressed by RNA-seq, and POGZ knocked down human CD34 derived early erythrocytes. And there were two that were standouts with regard to expression pattern. This is one, REOC3. You can see here it's expressed only during erythroid differentiation. I said, okay, so let's go for this one, because maybe this is something we can make a pill for. And when she makes a smear for REOC3, you can see here, this is a scrambled smear. This is beta globin, and this is gamma 1, gamma 2. So not that much in this culture, gamma 1, gamma 2. But when she uses this smear against REOC3, you can see beta goes almost completely away and is almost completely replaced by gamma 1, gamma 2. So this is a switch from adult to fetal, and it's nearly complete. This is by qPCR. So this is the kind of thing that I think we should be targeting with a pill. So I'll summarize by saying, whole genome therapy works for sickle cell disease. Most patients can be cured this way, even with a half match. So almost everyone has a donor. Gene therapy trials are now accruing, and I think I've shown you at least a half a dozen where it's working. We need to do better. We need to, think about risks, mitigate risks. Also, we need to think about ways to do this in vivo, because it's cumbersome to take these cells out and do transplants. And finally, expanded access should help to improve the outlook. We have both the Cure Sickle Cell Initiative and the Bill and Melinda Gates Foundation, pushed by Francis to partner develop gene-based cures for sickle cell disease, and this has really been pushing the in vivo gene transfer. So I'll acknowledge the crew here and pass the baton.
Thank you so much, John, and we'll turn it over to Tim.
Fantastic talk. So it's my huge pleasure to follow John in this last session of the Bold Prediction series. My name is Tim Yu. I'm a neurologist and geneticist at Boston Children's Hospital. And let me get my slides up for all of you. So for my talk, it's a treat to be asked to give this presentation. I get to speculate a bit. I get to talk a bit about some work that we're doing that relates to some of these predictions. I get to tip my hat to a lot of exciting work happening in the field from many others that I think are early indicators that this prediction here, like John, I think that this prediction here might not have been bold enough. I'd very much like that to be true, but I'll try to convince you of it. As we said at the outset, this bold prediction number 10 is that breakthrough discoveries will lead to curative therapies involving genomic modifications for dozens of genetic diseases. And a brief outline of my talk is to tell you, 1, what makes this prediction bold, 2, how this prediction can come true, and 3, how will we make this work and hopefully prove it to have not been bold enough. So let's start with that first. So let's start with some sobering statistics. This is a slide that I put together some 4 years ago using pharmaceutical industry statistics from 2014, illustrating the point that therapeutic curative development is dauntingly expensive. In these statistics commissioned here and referenced in the NIH Director's blog in January 2014, the average industry cost per new drug approval was a daunting $2.6 billion, with the time from target discovery to FDA approval of a drug a daunting 14 years. Worse, but not listed on the slide, is the fact that 85% of leads actually did not make it through from beginning to the end, adding to the overall cost of each approval, successful approval. And this indicated that developing therapies has traditionally required incredible investments of money and time with no guarantee of success at the end. Now, with respect to this prediction, add to that the fact that the number of genetic diseases, partially because of our own success, keeps rising. This slide, borrowed from Chris Austin, former director of NCATS, and sourced from the OMIM, shows the number of distinct genetic diseases with a known molecular basis. And as of 2020, we were up to nearly 7,000 of them. And as a sobering checkpoint, if you ask how many of these actually have a targeted therapy available for them, there are only 500 of them, just a tad over 7%. Worse than that, the new genetic diseases that we are finding, of these, the great majority of these are incredibly rare, especially in my specialty. I think neurology is perhaps one of the last holdouts where many of the genetic etiologies have been only recently become clear. And the great majority of new syndromes that are being described, of course, it's early days, we are finding that they're incredibly rare, meaning that there are much, much less than 1,000 individuals, more like a few dozen individuals described. And what that means is that the prospects of accumulating enough natural history information, also accumulating the number of patients necessary to justify commercial investment, is another daunting piece of the puzzle. So all of this points to the fact that as we talk about the remarkable advances that have been catalyzed with the Human Genome Project, that to actually execute one of the promised fruits of the Human Genome Project, which is genomic medicine, we have to mind the remaining gap. Genomics has hugely shortened the discovery odyssey and increasingly the diagnostic odyssey, but treatments remain few and far between. So how is this going to possibly change in the next, by the year 2030? Well, the good news is that it's already beginning to. And let me explain to you how, in concept, this prediction, I think, can come true, and why, again, it could prove to be not bold enough. To illustrate this idea, first, I have a slide showing you traditional drugs that we've used in our armamentarium for decades: Tylenol, penicillin, Taxol, metformin. These are all small molecule drugs, painstakingly either purified or synthesized from various sources. And each one is unique, like a snowflake, where it has its own shape, its own structure, its own particular quirks, its own biodistribution, its own potential safety and safety profile, its own PK. And this has traditionally made efforts to develop cures based off of small molecule technology really quite painstaking. Thankfully, we are experiencing now a new wave of platform-based drug modalities. The tools for drug development are getting better and better. It's increasing the efficiency of drug development. And that increase in the efficiency allows companies to go after rarer and rarer conditions with fewer and fewer resources. Some of these platform modalities are shown on the slide here: gene therapy, antisense oligonucleotides. I'll spend the bulk of my talk talking about those two. And at their heart, what they encapsulate is the promise of this: a transition from snowflakes to genetically programmable drugs. This is an image taken from a review that I like from my colleagues, some colleagues at the University of Massachusetts, John Watts and Antonia Kvarvova. And these show how traditional small molecule drugs have a chemical structure which confers upon it two properties. And here we'll throw a couple of vocabulary words at folks. There's the ionophore and the pharmacophore, which refer to properties of the drug that dictate for the ionophore its uptake, distribution, and safety, or for the pharmacophore, its biological target. And the point is that these two properties come as a unit. for small molecules, that their drug properties of this compound and its targeting properties are intertwined. In contrast, the new wave of platform-enabled genetically targeted drugs represent informational drugs. Informational drugs are those in which these two properties are actually separable. There is a dianophore, which modulates its drug properties, and a sequence, which modulates its biological targeting. That sequence might be a guide RNA in the case of CRISPR, or it might be the sequence of the molecule itself, as in an antisense oligonucleotide, or an mRNA molecule, like a COVID vaccine. And that these two properties, the drug properties and the targeting, are therefore separable. There's an asterisk next to that because they may not be perfectly separable, but they are much more separable than small molecules. So this concept of modular drug development, also going by other catchphrases like platform-based drug development, programmable medicines, informational therapies, this is what I and many other people are excited about. To make this real, let me give you an example of an informational therapy. It's one that's near and dear to my heart. It's called a drug called Niacin, and its sequence is shown right here. Some 2 years ago, my group published a paper in the New England Journal describing a very unusual and fortuitous collaboration that evolved based around developing a drug for a single patient. And this is a story that's been widely discussed in the literature. For those of you who don't know, it began with our meeting a 6-year-old girl with vision loss and neurologic decline, who was diagnosed by her expert clinician, Dr. Austin Larson, with a really devastating disease known as CLN7 Batten disease. This is a rare disease reported in the literature in some 70 patients total, and it causes progressive neurodegeneration and, unfortunately, death at a young age. And there were no treatments available at the time of her diagnosis. This is a genetic condition. It's usually caused by recessive mutations in a critical lysosomal gene. And when we offered to help with whole genome sequencing, we found an unusual finding, a cluster of mismapped reads in the sequence of her CLN7 gene deep within an intron. And that cluster of mismapped reads eventually led us to recognize that she had suffered an unusual mutation, the insertion of a mobile DNA element, or a retrotransposon, deep within the 6th intron. And the insertion of this heterologous sequence was creating a new splice site that actually truncated the gene and disrupted the function of this transcriptional unit. In a story that has, as I mentioned, received a lot of attention, we were fortunate to realize that there was a scientific possibility here, which was to develop a customized medicine to silence this abnormal splicing event and to rescue gene function. And with the help of some amazing collaborators, that's what we set out to do. This whole concept was based off of a technology called antisense oligonucleotides, whose most famous initial example is a drug called nusinersen for spinal muscular atrophy. Nusinersen is an 18 nucleotide synthetic RNA that is simply dissolved in artificial CSF, or not much more than normal saline. It's given by spinal tap to infants with this terrible neurodegenerative disease affecting the motor neurons. And there, when given to these infants, it is capable of binding and restoring normal splicing of a critical neuronal survival product called SMN2. And by altering the normal splicing of SMN2, a gene which is present in all of us but not typically used, this ASO drug was capable of restoring function of this SMN2 and rescuing the phenotype in patients with SMA. It was based off of this platform technology called antisense oligos, or ASOs, which are short snippets of synthetic DNA or RNA, typically 15 to 25 bases long. They're easy to manufacture. They bind to their targets by Watson-Crick base pairing. And over several decades, work by many folks, including some of those on this slide, have led to the generation of multiple versions of chemical modifications that give them very good drug properties. They're well absorbed, well distributed, quite stable, and quite safe. And this drug platform is seeing increasing use in clinic to either boost gene expression by modulating gene splicing or to shut down genes by targeting RNA for destruction. Reading about this technology, even though we had not previously worked on ASOs before, we were able to adopt these tools and with help from many collaborators in the field, as I mentioned, develop a customized oligonucleotide specifically for our patient. Knowing her specific mutation, because we had done whole genome sequencing, we could target the splice site that had been created by her unusual mutation, design an ASO complementary to it, test it in her own cells, and then validate its function to rescue the health of her own cells, conduct safety studies, and then get permission from the FDA to proceed with an investigational treatment for her within just 1 year's time. This effort with the timeline shown here points out how far advanced these tools have come. From the point that we actually identified her mutation to the first dose was just 10 months. From the point that we actually demonstrated that it was causing the splicing to the first dose was 8 months. And actually from the point where we first had our candidate lead, where the functionality of our lead molecule was demonstrated in a dish, to the time that she was actually dosed, that was a previously unheard of 3 months' time. And this effort, which we talked about at the American Society for Human Genetics in 2018, led to an upsurge in interest in this concept of programmable medicines. With our template, nusinersen, for spinal muscular atrophy, the 18-letter sequence shown on top. With our drug for our patient, a slightly longer sequence using exactly the same chemistry, but just a different order of letters for our patient, resulting in a different programming, so to speak, that proved to be of some benefit to our patient. This effort has been followed by other efforts from our group and others. Here is an infant with a condition called ataxia telangiectasia, a fatal genetic disorder that we met shortly after meeting our index patient. This baby girl had an abnormal newborn screen, so she was actually diagnosed with this condition at birth. And this is, as I said, a progressive and ultimately fatal condition that leads to degeneration of the cerebellum. Infants with this progress to be wheelchair bound by age 10, and it's fatal, unfortunately, by young adulthood. There are no available treatments for this disorder, but when we met her, we studied her mutations and realized that one of her mutations created a new splice site in the causative ATM gene. It was very similar to that which we found in our Batten patient. And we were able to, in partnership with the AT Children's Project and many collaborators shown here at our institution and others, develop an investigational drug for this patient in just 18 months' time. And we were able to initiate injections for her under appropriate FDA guidance at age 2. This effort, which is still ongoing, has also been paralleled by other efforts that have demonstrated the feasibility of using platform technologies like ASOs to bring to bear treatments for patients with short turnaround times. In 2019, a patient named J.C. Hermstadt, who had a severe genetic form of ALS, received an investigational ASO therapy championed by Neil Schneider, Bob Brown, and John Watts. This therapy for the FUS form of ALS has turned first from a single patient trial into a 9-patient trial, and now is enrolling into a phase 3 trial. My colleagues, Bob Brown and John Watts, also just actually first in preprint last year and recently in Nature Medicine this year, described development of another individualized ASO for another genetic form of ALS, C9orf72 ALS. And these examples are just some of many other efforts that are underway led by academics like myself, foundations, some even led by patient families working with CROs. And these efforts illustrate the advantages of using, in this case, antisense oligonucleotides to speed development, listed here. The fact that they're rapidly customizable, the fact that they're simple and inexpensive to manufacture, they're relatively easy to deliver, and they have a growing safety and efficacy record through trials like this, plus many other more traditional industry supported trials. What these experiences illustrate is the importance of the need to nurture therapeutic platforms to meet this bold prediction of 2030. That we are experiencing an upsurge, the emergence of these therapeutic platforms with technologies like antisense oligonucleotides, where different drugs can be targeted to different genes, even to different individual patients by changing their sequence. We are seeing a renaissance in gene therapy with AAV gene therapy taking the lead for AADC deficiency, different vectors, promoters with different payloads targeting AADC deficiency, spinal muscular atrophy, giant axonal neuropathy, and retinal dystrophies, and many other technologies that share these same platform characteristics. Phosphomorpholinos, like the remarkable exon skipping therapy led for DMD by Sarepta, siRNAs, mRNA therapies, and CRISPR genome editing. All of these are poised to benefit from this separation between delivery and payload. How does this separation work? Well, for ASOs, it's relatively simple. The formulation is simply dissolving them in the case of central nervous system administration, artificial CSF. So you keep the chemistry the same backbone, the nucleotide modifications, and you vary the sequence. For phosphomorpholinos or siRNAs or mRNAs, these are slightly more complicated. You need to formulate these with chemical modifications for stability. You may need lipid nanoparticles or cell-penetrating peptides, but the coding may be optimized, but hopefully then stabilized and standardized. And again, the sequence may be varied. Finally, for gene therapy or ultimately in vivo CRISPR editing, there is the same prospect to keep the viral vector, the promoter, the regulatory elements as constant as possible, depending on the target you're looking for. And again, varying the payload, varying the sequence, the cDNA or cDNAs that you are delivering, or the guide RNAs in the case of CRISPR that you are editing. And I will note that CRISPR edits, guide RNAs are simply a form of an antisense oligonucleotide. Well, how well is this going to work? Are we really going to make it in 10 years? And I do want to highlight that this is not a trivial problem. And the main issue in my mind, like as I say in real estate, location, 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 the main issue here is delivery, 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 delivery. Are we going to be successful in finding the right wrappers for PMOs, siRNAs, mRNAs? Will we have the right serotypes of AAVs or a next generation virus that will come down the road to get it to the right location at the right dose with the right duration of action? Will it work immediately or perfectly? No. Viral vectors may predictably remain finicky. The payloads, not every sequence within a class will behave exactly the same. This is proving true for antisense oligonucleotides as well. But with time, if we do this right, we'll be able to learn to manage these. And if we do this really right, we'll even be able to predict them. The fundamental sea change that this new science dictates is a change in the way that we do business. In small molecules, any two drugs are more different than similar. Any two snowflakes. It makes sense, therefore, for these drug efforts to develop largely in silos, where the efforts from one are not easily generalizable to another. And that has dictated many of the ways that our community and our society does business and how our companies do business. But next generation drugs hold out the prospect that any two drugs will be more similar than they are different. Instead of data silos, maybe what we need is something more like data potlucks. Everyone gathering around the table to share what they've got and to benefit the next effort. We are already beginning to see consortia develop along these themes. I'd like to highlight in particular Ph.D. and BGTC. The Bespoke Gene Therapy Consortium is a remarkable coalition that was just announced a few months ago, including NIH, industry players, the foundations for NIH, and many, many others, companies and foundations from the community. And I think Peter Marks, the director of CBER, set out the goals, set out the aims very, very clearly. We could get more diseases more quickly. We could get to more diseases more quickly if we had higher throughput by leveraging something that's inherent in this class of products. That's what gets me interested in this as a regulator. And Bespoke Gene Therapy Consortium, along with the PAVE-GT program sponsored by NCATS, which was a critical pioneer paving the way for the BGTC, is focused on standardizing AAV manufacturing, safety assessments, modernizing clinical trials, developing master protocols using bucket and umbrella designs, consolidating regulatory filings, stepwise systematically increasing the efficiency of the scientific, logistic, and regulatory process. I'd also like to highlight NINDS, which recently launched the Urgent Network. This is a network devoted to the preclinical and clinical coordination of gene therapeutics, genetic therapeutics for ultra-rare neurogenetic diseases, with thanks to Nina Shore, Chris Boshoff, and the late Anne-Marie Broom. Urgent was designed from the ground up to provide support for gene-based therapies for ultra-rare neurologic diseases affecting fewer than 1 in 50,000 people. And its vision is to democratize access to gene therapy, to facilitate the study, understanding, and treatment of patients, and to iteratively improve the efficiency, cost, and tolerability of these gene therapy efforts. In our own work, I see that we are working hard on antisense oligonucleotides at the moment. I see this as an early exemplar platform for testing out some of these new structural ideas. And like in gene therapy, this is a space that has seen multiple parties step in to also contribute, including the charitable N. Lorem Foundation, founded by Stan Crook and Frank Bennett, devoted to developing medicines for free for patients with ultra-rare conditions. And our own effort among the academic community to form the N of 1 Collaborative for Individualized Medicines. This is an international hub of some greater than 100 participants from academia, including patients, governments, and biotech companies as well, with the mission of making individualized genetic medicines safe and accessible. The goals here are to develop the best practices for the clinical conduct and delivery and evaluation of patients for efforts like Mealison and similar. And the types of data sharing that I think exemplifies what is going to be more and more of the expected route to come with this type of technology is shown on this slide, contributed by my good colleague, John Watts, one of the steering committee members of the N of 1 Collaborative. Within ASOs, there are now agreements in place to share in vitro cell culture screen data, animal safety toxicology data, and clinical experiences that will contribute to the amassment of data that can be then mined to help better predict and manage the remaining uncertainties that come with platform medicines. The types of efforts that are required for this data sharing include the development of community accepted standards for data interoperability, to enable groundbreaking statistical and computational methods to be applied, and to encourage data stewardship models that ensure open science and reduce data access burdens. If any of that sounds familiar, it's because it's shamelessly plagiarized from the 2020 NHGRI strategic vision as highlighting the accomplishments of the Human Genome Sequencing Project, now just harnessing those for therapeutics. And so some of the goals in this collaboration is to establish reference sets of safe versus toxic ASO sequences, develop cell-based predictors of toxicity or off-target effects, and ultimately to develop AI and computational predictions of tolerability. On the home stretch. So I'm going to reprise my intro slide. One of the daunting and bold aspects of this prediction is the current standard for how much it costs to develop a drug, $2.6 billion in 14 years. I'll just point out, though, that if that number sounds kind of familiar, it actually is very, very similar to the cost of the Human Genome Project. When it was launched in 1990, it took 13 years to complete, and it was completed to the tune of $2.7 billion. Now look how far we've come. In 2022, sequencing a human genome has become democratized. Almost anyone, it's not enough, but getting there. Medical patients who have need can have a human genome sequence for $1,000, and it can be done in specialized with special experts like Stephen Kingsmore at Rady Children's Hospital in even less than a day. So the question is, can we do the same thing for therapeutics? The cost of a new drug approval in 2014, what is the fold reduction that we anticipate being able to see in 2030? And this democratization of the DNA sequencing process led to a 1 million-fold reduction in costs and the potential of genomes for all. Can we do this again? So I'm going to close here, taking the liberty of making my own variants on the bold predictions, my own bold predictions for human genomics by 2030. And with your some latitude from the audience, I'm going to throw out there that I think that open standards and consortial models for programmable drug development will drive down costs by 1,000-fold, democratizing access to treatment for hundreds of not just common, but also rare diseases. Number two, I think that we're beginning to see this already. Individualized treatments will impel modernization of regulatory frameworks and clinical trial designs, challenging, in some instances, what we consider a drug versus a medical intervention. Third, and I have to get a newborn sequencing here, a brief mention. Newborns will be sequenced at birth, allowing interventional genomicists to offer customized, just-in-time treatments targeted to their personal mutations. Thank you very much for your attention, for your forbearance with my own bold predictions. I'd like to thank many people who've been involved in conversations along these lines in this work over the last 4 or 5 years from my group and extending to many others. And I have more detailed acknowledgements to members of my lab that contributed to the scientific work I described, the N of 1 Collaborative and other critical collaborators. Thank you very much.
Well, thank you very much, Tim, and again, thanks to John. And I must say that this has been truly an inspiring pair of talks. So Tim has sort of thrown down the gauntlet, if you will, with regard to his bold prediction, and John has told us that we're a little bit wimpy in terms of our NHGRI bold prediction, but may I just ask John if he has some bolder predictions for us in NHGRI, whether dealing with rare diseases or more common conditions such as sickle cell.
Well, so, I mean, my own bold prediction is that we'll solve in vivo gene editing for diseases, not like liver diseases. One of the questions that's already come up is about liver disease and in vivo gene editing. I think most of the things that we try to put in the body wind up going to the liver. And for those of us that are trying to target bone marrow, for example, that makes it very difficult to get those reagents to the bone marrow. And so, if we really want to go bold and fix a disease that has a huge impact on the globe, like sickle cell disease, we can't do it in the way that we're doing it now. It's not bold enough to say we fix sickle cell disease with gene addition, because we can only do that in places that have the tools to put that together. So that's not really bold. What's bold is to say we're going to cure sickle cell disease, not here where there's less than around 100,000 people, but where there are millions and where we don't even know about most of them because they die before they're 5 years of age. So to be really bold is we have to develop screening programs where diseases are prevalent, and then have the kinds of therapies that could provide for cures where they are. So, my bold prediction is that we will reach that for sickle cell disease with in vivo gene editing.
Okay. All right. Well, that certainly is bold. I will say that. So one of the concerns that we have, of course, in NHGRI and for the NIH writ large is the question of what can we do programmatically as funders of biomedical research and these kinds of programs to make the field move ahead. What are the most efficient things that we can do as a funding agency to enable some of these advances? Tim, do you want to start out and then John?
Oh, I would love to. So I think that as we think about what is possible with the technologies that we've got, we have to think, is the scientific possibility well matched to the system that we've designed to deliver on this promise? And we have set up a system, for instance, where appropriately so, that drug development is really fueled by tremendous industry investments. And I think that's very, very effective when it comes to common diseases, because there is a write once, read many times ability there to take an investment and then develop a therapy and then be able to deliver it at some reasonable profit to many, many people. And I think it's more challenging in rare diseases. And I think that as we think about now extending what's scientifically possible with the systems that we've got to fund the work, I think that we'll find that NHGRI and NIH, I would love to see our government agencies work hard to try out and prototype some other interesting models to go after these conditions that may not be right for commercial investment. These are bold ideas. They're risky ideas. They may fail. There are hazards of these platform technologies that have yet to be discovered. But there isn't necessarily help coming from other corners. So I think that efforts like the ones that I mentioned, at the BGTC and Urgent, are examples of the types of programs that I would love to see more of as an academic who sees this as a really productive unmet need that we could devote our resources to.
So sort of proof of principle kind of work that then could lead to investment in the ones that are successful. Okay, John, do you have any thoughts with regard to what the NIH or NHGRI in particular could do to?
Well, I agree with what's already been said. And I think NHGRI and the NIH in general have been doing just that, leading, putting, people together, like in spaces like this or in meetings where, okay, here's the problem that we want to try to fix. Let's get all the people together. Let's figure out what the most important steps are. And then figuring out funding mechanisms. I mean, for example, I was a part of Francis's efforts to get funding for sickle cell disease, for example. And once people understood that the real push was to develop in vivo gene therapy, it was such a moment of science fiction, actually, when we were talking about this. And now we have, just a few short years, examples of it working in humans, and other examples of it, looking pretty promising and ready to roll out for human clinical trials. So, knowing the landscape and knowing what's important to develop next, I think is what the NIH does best and should continue to do, so that we get funding to the right areas and to the right kinds of projects to move things forward. I do think we lose a little bit on the investment side by not claiming any part of what what we develop that then goes to industry. But that's another question. A better way to partner, I think, would also be helpful. And I see there's been some movement on that, but that could also be of help.
Yeah, I agree. Well, perhaps because it is 4:15 and we only have 15 more minutes, at least of our allotted time, I'm going to turn things over to Chris Gunter, who I know has been monitoring the Q&A function in Zoom and probably has, at least 100 questions that we'll need to get through in the next 15 minutes.
Exactly. This is the lightning round part of the talk. And thank you, everyone. Dr. Tisdale has already answered a few of the questions in the Q&A box, so you should be able to see that if you're interested. So I can answer one of them too, which is that someone asked, when can we see this lecture again? Because it was so great. And the answer is that our wonderful communications and IT teams usually put these up within a week after they have been put up. So I would estimate around then, and thank you to them. So one of the first questions that we got was about more focusing on CRISPR technologies and what is the role of CRISPR technology in the future of these gene therapy applications? And secondly, does CRISPR technology carry any higher inherent risk of causing chromothripsis or off-target effects, for example?
I can start that one. I think when we think about new technologies, we're always, of course, appropriately worried about off-target effects, and we should be. We should be monitoring for those. And it would be best even if we had a central repository for data where we could all mine the data and make sure that we're all thinking about this in the same way. But let's not forget that, a lot of the things that we do, for example, we're giving busulfan conditioning for our patients before we give them their genetically modified cells, which does, a lot of DNA damage, let's face it. And if you were to quantitate the DNA damage done by chemotherapy versus by, CRISPR, which is, following the rules, so to speak, it would be infinitely less, I think, by this technology, if you were to predict, than by, for example, the chemotherapy that we use to condition the patient. But, once we start thinking about in vivo gene therapy, and I think this is what the question was about, we have to think about not only off-target effects, but off-tissue effects. So are we getting into the, germ cells, for example? So that's another big question that we have to think about when we start squirting reagents into humans and expecting them to fix their bone marrow cells or their liver cells or whatever it is we're doing. We may have effects in other places that are not off target, but are off tissue. And we have to think about those as well.
Tim, do you want to add an answer to that?
I think it's interesting to think about the roles that CRISPR will play in going after common mutations. I think that many of the current applications have used really elegant biological insights like the BCL11A pathway to knock out a critical switch that's built into the system to allow us to affect a change that is of therapeutic value. In fact, the spinal muscular atrophy SMN2 situation for ASOs is exactly the same thing. A hidden switch that's built into our genomes, evolutionarily set there, almost by purpose, of course not, that we can exploit using a single modification that you can study and use over and over again. I think the interesting challenge is to think and envision a time when we can use CRISPR to reverse not just the inherent biological opportunities that we all have, but the specific mutation, the de novo mutation that arose in just my child that broke a splice site. Can I change that GT that was splice donor that was mutated to something else back into its original form? And that will be sort of the ultimate demonstration and will require the most amount of ability to predict off targets and screen for them using whatever in silico or in vitro tools are necessary at the point. I think that that's sort of the ultimate goal that would unlock treatments for almost any arbitrary condition. And if we can hit that, then we'll see a huge inflection point. And then the prediction of dozens will be a huge underestimate. It'll be hundreds. If we can't hit that, then we may have more trouble.
Yeah, absolutely. So, Tim, I'm going to put a question to you, which John has already answered. It's from our newest full-time faculty member, one Francis Collins, and he is asking to look at the best long-term outcome for transplant or gene therapy in sickle cell. It would be good to carry out therapy before organ damage and bone marrow P53 mutations appear. What will it take to get approval of children? And I know that was mainly aimed at John, but I don't know if you want to say anything more about what it might take to get approval for similar therapies that you work on in children.
Oh boy, thank you. I'm honored to take that question. I think that children pose a really a special use case, and I think that on one hand there is a lot of traditional appropriate safety measures that are put in place before advancing treatments into children. The idea of testing adults first whenever possible is ingrained in our sort of regulatory and clinical and ethical training. But I'll just comment that actually poses some significant challenges for the types of therapies we're dealing with now, because the types of therapies that are being proposed might be specific to a very, very small number of individuals, maybe 1,000, maybe 100, maybe one. And so what are the appropriate guardrails and ethics for giving those to an adult volunteer first? It doesn't have the same ring to it. So that, coupled with the fact that so many of children, it's a silent epidemic because so many children with severe genetic illnesses are dying at a young age in the first few years of life. They don't count to the statistics for later age adults. I think that it is forcing us to reevaluate that and to hurry things for at least the severest conditions to kids much more quickly.
Yeah, thank you. That's definitely a theme I think we're all interested in. And leading to that is we've gotten a separate question, which is a little bit similar. How do we encourage more researchers to focus on the type of therapeutic research discussed today? Are there any, a related question might be, are there any big hurdles that are in your way that you wish someone could just make go away?
Well, I think I might jump in and just say that it's always good to encourage researchers to follow what drives them. Let's face it. Many would have said, who cares how bacteria resist viruses? Let's work on human disease, and we wouldn't have CRISPR. So we need this kind of curiosity-driven science to feed those of us who are interested in developing therapies for humans. We need all of it. So I think what we really need to do is, to encourage great science. I don't know if you have anything else to add to that.
You said it perfectly, John. I think that's one of the driving philosophies around our training, and you never know where your curiosity will take you. I think also at the same time, for those of us who, when you are committed to a particular translational cause that you've decided is your passion, then I think that the concept of flexibility and looking for asking yourself the hard questions for how new capabilities change the way you think about business as usual, whether those are questions to the seeking funding from industry versus NIH versus other sources, philanthropic sources, whether it's questions about appropriateness of starting first in human therapies in children, or other really challenging questions. I think that the investment that NHGRI in particular has put into ELSI is a good way to inoculate us against being stuck with old and possibly ideas that need modernization.
So if the two of you were talking to trainees, which I'm sure you do very often, who said, I want to do a lot of what you're doing, I want to be involved in this bold prediction and making this happen, what would you tell them? Is there any course that you wish you had taken that they should really take? Uh-oh.
I'd encourage them to listen to every seminar in the series. I really hugely enjoyed all of the talks. Really, the invitation. You don't get an invitation as a scientist to go out on a limb very often and explore concepts and usually we're trying to be more conservative than that. So I think that this was a great series and a great place for trainees to start and stretching themselves a little bit. How about that?
Thank you. And it also sounds like maybe taking a class in ethics or thinking about something like that would be useful based on what you just said. Yeah. John, go ahead.
Yeah.
Well, I was going to say, that to think broadly in the beginning while focusing, so it seems, it seems like two different things, but I think, to think broadly while focusing really help to even narrow the focus and to be more successful. And with trainees, I like to encourage them to have a certain project. I mean, we always, have something in mind that we'd like to see done, but, once they start running with their own ideas, it's always much better. A way to enable them to think broadly while focusing, I think is important.
Great. Okay. I have 1 minute left before. Oh, go ahead, Tim.
I was also just going to, on that same theme, thinking broadly, I think cross-training is just increasingly so valuable. And whether it's biology and engineering, if it's genetics and computer science, statistics, and you name it, or maybe it's taking a drug development course and seeing how science is practiced in industry. I think that each of those gives you opportunities to learn how something is done and ask yourself, why is it done that way? And is there a way to take something from here and bring it over there and mix it together?
Yeah, absolutely. And we had one question that was thinking about big data. And I think, Tim, you answered that as well in your talk, talking about big data. But I think that's part of what you just said there is learning how to deal with big data as well would be useful for trainees, it sounds like.
Increasingly so, yeah.
All right. Well, I'm going to turn things back over to Dan. Thank you both so much. We really appreciate you speaking today. And Dan, take it away.
All right. Well, Chris, thank you very much. And thank you, Chris, for your moderation of those questions, which were really fantastic questions. And I do want to thank John and Tim, both of them, for spectacular talks and for really getting us to think both deeply and broadly about these questions. And that's really, really important. And part of the whole point of this is to really get us to expand our horizons and to imagine what things could be like 10 years from now, even if we can't exactly predict it. And I would like to also bring this series to a close. I think it has been a fantastic experience over the course of the last 11 months. I would certainly encourage those of you who have not had a chance to partake of some of the other sessions to do so. And the fact that we have these accessible through the NHGRI website and also through YouTube makes it easy to do. And certainly I would encourage everyone to do that as well. So thank you all very much. Thank you, the participants. Thank you, those who have tuned in today, and thank all of you who will tune in over the course of the next few months to a year or whatever. So with that, I will call it to a close.